My name is Matt Kozlowski, and I'm an assistant professor in the Department of Statistics at Colorado State University. Today I'm going to present a Bayesian joint model we developed to simultaneously identify clinical covariates associated with the human microbiome and predict a phenotypic response using information contained in the microbiome data. Here is an outline of my talk. I'll first introduce microbiome data and some of the current methods researchers are using to analyze these data and their limitations. Next, I'll present our Bayesian joint model, which we apply to microbiome data collected on individuals in the Houston area. Then I'll conclude with some final remarks. The human microbiome is a community of microorganisms that live on and inside of our bodies. The goal of human microbiome research is to understand the role of our microbial communities and how they interact with their host, respond to their environment, and influence disease. One of the primary research questions in human microbiome research is the feasibility of designing interventions that modulate the composition of the microbiome to promote health and cure disease. There are two main objectives researchers pursue to address this research question. The first is identifying factors that characterize the compositions of the microbiome. For example, we might be interested in the relation between diet and microbial abundances. The second objective is predicting biological, clinical, or experimental conditions using microbial abundance data. For example, we might be interested in investigating how we can predict BMI using the microbiome data. There are a variety of challenges we are faced with when modeling human microbiome data. The first is that they are compositional in structure. As such, the relative taxa proportions sum to 1, which makes for a dependent structure among the taxa counts. Microbiome data are high-dimensional as we are often dealing with large covariate and taxa spaces. They're over-dispersed due to large within and between subject variability, and they're oftentimes zero-inflated, as we often observe lots of zero-read taxa counts. For the first objective, there are numerous regression models available for relating covariates with multivariate count data. For example, the Dirichlet multinomial, which we use in this analysis, the Dirichlet tree multinomial, as well as the negative binomial model. In order to accommodate the compositional structure of the microbial data in Objective 2, researchers apply log ratio transformations, such as the additive and centered log ratio transformation, as well as the isometric, often referred to as balances, which I'll define later. After performing these transformations, researchers can use traditional analytical approaches for analyses, but inference changes based on the transformation applied. In practice, researchers typically model these two objectives separately in two-stage approaches. However, this underestimates uncertainty in the count data when treating them as fixed, when predicting a phenotypic response. And additionally, we're not using all the information available for inference, which results in overconfident and less accurate predictions, which we show in simulations. In order to fill this gap, we develop a Bayesian joint model to identify clinical covariates associated with microbial data and predict a phenotypic response using information contained in the compositional data. Here, we model the relation between clinical covariates and microbiome data using a Dirichlet multinomial regression model. However, instead of integrating out the taxa proportions, we sample them, transform them into balances, and use them to predict our phenotypic response in the second layer of our model. As such, the taxa proportions serve as the shared parameter in our joint model. To reduce the dimension space of our model and simplify inference, we assume a sparsity-inducing priors at both levels of the model. And additionally, we take advantage of a data augmentation technique for sampling the taxa proportions, which greatly reduces the computational demand of the model. In order to identify clinical covariates associated with microbial data in the first part of our model, we assume that the taxa counts, Zi, follow a multinomial distribution where Z.I is equal to the sum of the counts over all taxa, and psi I are the taxa proportions, which are defined on the j-dimensional simplex and are assumed to follow a Dirichlet distribution with concentration parameters gamma. We introduce covariates in the model using a log linear regression framework, where lambda ij is equal to the sum of a taxon-specific intercept term, alpha j, plus the sum of a p-dimensional set of clinical covariates and their corresponding regression coefficients. To perform variable selection, we assume spike and slab priors, which are characterized as the mixture of a slab distribution and a point mass at 0. Here, we assume that these zetas, which can take on a value of 0 or 1, represents a latent inclusion indicator for each one of the regression coefficients. If zeta is equal to 1, then the corresponding covariate is included in the model. If zeta is equal to 0, then the corresponding covariate is excluded from the model. Typically, we assume that these inclusion indicators follow a beta binomial distribution where A and B control the overall sparsity of the model. And again, we use this prior specification to help us identify which clinical covariates are associated with taxa proportions in the first stage of our model. In the second layer of our model, we regress a phenotypic outcome Y on a set of balances constructed using the psi estimated in the first layer of our model. Here, alpha naught represents an intercept term. Beta m are the regression coefficients corresponding to each one of the balances in our model. In terms of balances, consider two non-overlapping partitions of psi positive and psi negative. Here we define the balances for these partitions as equal to the square root of the product of the number of taxa in each partition over their sum times the log ratio of their geometric means. In practice, the balance structures could be defined using a phylogenetic tree which incorporates the evolutionary relations among the taxa. Alternatively, we use sequential binary separation, which is visualized in the tree graph above, since prediction performance is agnostic to the ordering of the leaf nodes. Again, we assume a spike and slab prior for the corresponding balance coefficients beta to induce sparsity on the balances for prediction of the phenotypic outcome. For posterior inference, we jointly update the regression coefficients and inclusion indicators to avoid reversible jump MCMC algorithms. Additionally, we marginalize out the corresponding regression coefficients for the balances for efficient sampling of their inclusion indicators. We apply a data augmentation technique for psi, which was motivated by techniques used in the Bayesian nonparametric literature space. As mentioned previously, this greatly reduces the computational demand of the model. In the next couple of slides, I will walk you through the data augmentation technique, which first reparameterizes the taxa proportion psi using the well-known relation between the gamma and Dirichlet distributions. Here, we still assume that the zi follow a multinomial distribution, but we'll replace the size with the Cij over Ti terms, where Ti is the sum of the Cij terms over all j for each individual. Next, we assume that the Cij's follow a gamma distribution parameterized with concentration parameter gamma and 1. The tricky part computationally is calculating the Ti to the Zi dot terms at every iteration of the MCMC algorithm. Instead, we replace the 1 over Ti to the Zi dot terms with the following integral, which uses the gamma identity, and introduce an auxiliary parameter ui, which follows a gamma distribution. As a result, we obtain closed-form Gibbs updates for ui and Cij, which results in a huge reduction in the computation time versus a Metropolis Hastings update for the psi terms. Once our MCMC chains have converged, selection is determined using a marginal posterior probability of inclusion, which is equal to the average of the inclusion indicators over all MCMC samples. If the MPPI exceeds a pre-specified threshold, then the corresponding covariate is determined to be selected. So this is a graphical representation of our full model. So here we're going to model the relation between the taxa counts and the clinical covariates using a Dirichlet multinomial regression model. However, instead of integrating out the taxa proportions, we're going to estimate them, transform them into balances, and then use them to predict a continuous outcome using the linear regression model. This model was motivated by our collaborations with Drs. Christy Hoffman and Carrie Daniel at Baylor College of Medicine and MD Anderson Cancer Center. In this study, stool and oral swab samples, as well as dietary questionnaires, were collected on 56 subjects. Prior to modeling, 16S rDNA sequencing was performed, and the resulting reads were clustered into operational taxonomic units and mapped via the Silva database. After pre-processing, 245 fecal and 185 oral taxa, as well as 74 nutrient food group variables, were available for inference. In short, the objective of our study was to identify clinical factors associated with microbial composition and compare the predictive performance of the model fit with the fecal versus the oral microbiome data on BMI. Throughout the model, we assumed non- and weakly informative priors. For both the fecal and oral models, we controlled for sex and age in the linear portion of our model. And also, due to sample size restrictions, we used the approximate leave one out cross-validation with the expected log point-wise predictive density in order to evaluate the predictive performance of each of the methods. So here's an example of the results provided by our model. This is a heat map of the estimated regression coefficients for the corresponding dietary factors and fecal microbial abundance associations. In the bottom right corner of this heat map, you see a strong positive relation between prevotellocopri and a bunch of sugary factors. This is interesting because in other studies, prevotellocopri has been found in higher abundances among those that were overweight and those with type 2 diabetes. Overall, we observed several associations with what you might consider a Western-style diet. This is interesting since the Western-style diet is known to be hospitable and supportive of the development of obesity and other chronic diseases. Lastly, we found similar prediction performance for both the fecal and oral microbiome models. Overall, we developed a joint model for simultaneously identifying clinical covariates associated with the composition of the microbiome and predicting a phenotypic response. We provide an integrated analysis of the relations between behavioral, microbial, and phenotypic measures. In simulations not shown, we found that our model was more accurate in terms of prediction versus the two-stage approaches, and additionally it was better at selection as well. For more details, this work was recently published in the Annals of Applied Statistics. And currently we're working on extensions of this framework for causal mediation analysis. Lastly, I would like to make a plug for a software package we developed to implement our proposed method. This package additionally performs variable selection for Dirichlet multinomial and Dirichlet tree multinomial models using spike and slab priors. And we also include alternative prior specification for inclusion indicators, such as the Markov random field priors with unknown or known graphical structures to accommodate functional relations among covariates. I would like to thank the NSF for funding this work, as well as our collaborators. Thank you.